Okay, guys, today we are going to talk about a structure called the cell, about the cell membrane. Now, let's remember, we're in biology class, and so when I think of biology, I think of life, and life starts out as a single cell. All right? And if I asked you guys, hey, I want to draw a cell, what would you tell me to draw? You would tell me to draw a circle or something along those lines. This structure right here, this purple line, this is called my cell membrane. Now, the cell membrane is super duper important. All cells have it, okay? So this is in all cells. All cells have this structure. And that makes sense because if I took away that purple line and I said, hey guys, right over here, what is this? What is this right here that I'm pointing at? Or what is this that I'm pointing at? You guys wouldn't be able to say anything because there's nothing there. But as soon as I draw this and I say, hey guys, what is that? Now you can tell me, it's like, oh, that's a cell. So you have to have a cell membrane to have a cell in the first place. It's kind of like a water balloon. If you don't have the balloon, then you just have a pile of water. So cell membrane is really, really important. It helps define my cell, and we'll talk about that when we talk about functions here in a minute. Now, cell membrane has two other names. His name is also phospholipid bilayer, and we'll talk about what that means. And then he's also referred to as the plasma membrane. Okay. Function-wise, let's talk about, we have a couple functions we need to do first, and probably one of the most important is that he defines my cell. And what I mean by defines my cell is he shows me, like he actually makes the cell. Again, if I drew a little circle like this and I said, hey guys, what's that? You're going to say a cell. But as soon as I take that away and I'm like, hey, what's this? You don't get to say anything because there's nothing there. So you have to have a cell membrane to have a cell. That's what defining the cell means. The next function he does is he's going to let things, and things can be different here and after a while, he's going to let things in and out. Because remember, this over here, this is my cell. So he has needs. Like he needs food, he needs water, maybe this guy needs oxygen. And if you look, so if food has to go in my cell, water has to go in my cell, oxygen has to go in my cell. But if you notice, all of them have to cross that cell membrane to get inside. So he has to let things in and out. And of course, because he's a living thing, he's also going to have to get rid of stuff. He's going to have to get rid of CO2. And he's going to get rid of waste. And same thing, so CO2 has to leave, waste has to leave, and you notice that they also cross your cell membrane. So that's a really important function, that he lets things in and out. Now the last function is the property the cell membrane shows. This is called semi-permeable. And what that means is that he only lets certain things in and out, right? Because maybe you have something really bad right here. Maybe this is a chemical. Well, hopefully that cell membrane will be able to keep that chemical out, so that way it doesn't make your cell sick or your cell die. So semi-permeable, that's a very important concept. That's a very important concept we'll talk about later too. All right, so let's talk about the structure of this guy, which means we need to go back to this whole phospholipid. So phospholipid. Well, let's dissect that word. Phos is referring to phosphate. And then lipid, well, think about what your lipids are made of. They're made up of glycerol and fatty acids. And so that's what I'm going to draw, is I'm going to draw this phospholipid. And of course, he is arranged as a bilayer, and bi we know means two, so we're going to have two layers of these guys. All right, first part, phosphate. So here's my P, and then my 4 oxygens surrounding them. So this is my phosphate group. Now, if we think about characteristics of him, we see that he has all of these oxygen and it has phosphorus. So this guy is very, very polar. And we know that if you are polar, that means you are hydrophilic. And hydrophilic means you love water. And so that's going to be important to us here after a bit. Then this is attached to my glycerol. So CH2, CH, CH2. And then we're going to hook these to an oxygen. And then to a C double bond O, C double bond O, C double bond O. All right, so this right here, this is my glycerol. He acts like a glue to attach the phosphate group to my fatty acid chains. And so then after that, now I get to, whoops, we're going to make it come off of this C right here. So this C has a fatty acid chain that looks like that. And this one has a fatty acid chain that looks like that. Now, we're not worried about the number of carbons and hydrogens in these right now. That's beyond the scope. But you do need to recognize I have two different kinds of fatty acid chains. So this is fatty acid. Let's talk about characteristics. We know fatty acid chains, these hydrocarbons, these are non-polar. And non-polar means they are hydrophobic, which means they don't like water. Now, if I look at them, I know that there's a difference between my two fatty acid chains. This one has a double bond in it. That means it's unsaturated, so it's liquid at room temp. And then since this one is straight, we know he's saturated and he is solid at room temp, which is really important to the functionality of my cell membrane. And we'll get to that here in a second. Now, that's my phospholipid, and it looks kind of scary because it's a bunch of bonds and elements put together. Normally, when we draw a phospholipid, there's my phosphate head, and then here's my fatty acid tails, and where they connect, that's my glycerol. So this is phosphate. These are my fatty acid tails, and then of course the glycerol is where they're going to connect. And that's how we'll draw phospholipids from here on out. Say you guys are responsible for knowing the structure of a phospholipid. You're responsible for knowing this structure, but whenever we draw it, we're always going to draw this squid or this fancy man, something along those lines. Now let's talk about the bilayer part. The bilayer is really, really important because if we think about our cell, right, here's our cell. And I always ask you guys, what are we mostly made up of? We know that we have water, which means we're also going to have water inside my cell. So my phospholipids, they have to be arranged as a bilayer like this because I have water on the outside of my cell and I have water on the inside of my cell. And the water needs to be able to interact with these phosphate heads because water, remember, he helps moderate temperature. He's going to be our universal solvent. So I need him to be able to interact with the water really well. I couldn't arrange my phospholipid bilayer like this because one side could interact with my water, but then this one could not. And of course, I can't arrange my phospholipids with the heads on the inside and the tails on the outside again because I have water on the inside and outside of my cell and they're not going to be able to interact with it. So it has to be arranged in a bilayer just like this because I need to have the phosphate heads interacting with the water and then these hydrophobic tails on the inside where they don't get to be in contact with water. Okay, so now let's go over why our tails are arranged as such. Because remember, this tail right here, the straight one, he's solid at room temp, and this tail right here, he's liquid at room temp. That will give my cell membrane fluidity. If you guys think about a bubble, you know how bubbles, like, they can bend and they can move and they can interact with their environment? It's the same thing with my cell membrane. I can't have it be a rigid structure. If I had my phospholipid with two straight tails, right? Well, if these are two straight tails, that means it's solid at room temp. And if I try to get, let's draw another one. So if I had my cell membrane like this with two straight tails, and I need to get something in my cell, he's going to get bounced back because this is solid, and I can't move through a solid, right? Like if you guys try to push your pencil through your desk, you're not going to be able to because it's a solid thing. So we can't have two saturated tails. If I had phospholipids with two unsaturated tails, well, remember, unsaturated, these are liquid at room temp. So these don't get to define a structure. I wouldn't have that circle for the cell membrane. Liquids fill the shape they're in. So if I put water in a square box, it's going to turn into a square. If I put water in a circle box, it's going to turn into a circle. And so I can't say, hey guys, I need you to stay in this shape when their liquids are meant to take the shape that they're in. So I can't have that, which is why I have to have a straight and a kinked tail, or a saturated and unsaturated, because this straight tail is going to give me some structure, and then that kinked tail is going to give me flexibility. All right, so now we can actually start drawing our cell membrane, okay, because we have other pieces in there too. So here's some phospholipids. There, we'll do 3 of them. Okay, here's some phospholipids. No, there's my pen. Okay, this is called my bilayer. Now this right here, just to reiterate, this is my phosphate head, and then these right here, these are my fatty acid tails. Now there are other structures in this cell membrane besides just phospholipids. We also have some proteins. So we have a protein that looks like this. His name is a protein channel, because if you look, he forms a channel from one side to the other. This is actually a type of integral protein, and integral just means it spans from one side to the other. So spans one side to the other. Now, for the protein channel though, his job is to let large things in and out. Because if I look, like let's say I have a tiny little oxygen guy. So here's O2. Oxygen's really tiny and he can fit right in between the phospholipid bilayer, no big deal. If I have a big dude that looks like this, maybe sugar, say sugar can't go through there because he's too big, but sugar can go through my protein channel. So I need my protein channel so I can let really, really large things inside my cell membrane. So let's draw some more phospholipids. Say they're getting a little funky. Let's do one more. All right, so now we have another type of protein. This protein we find only on one side. Well, think about what you call your side vision. This is peripheral. This is a peripheral protein. And peripheral proteins, what they do is they act like an ID badge. If we're walking through the school and you see somebody you don't know, normally you look to see for the ID badge, so that way you know they're supposed to be there, or you look for the visitor badge. This is how cells identify self. You guys have heard of autoimmune diseases. You've heard of transplants not taking. That's because those things don't have the right ID badges. And this is how we also identify bacteria that are in our cells. Because once a bacteria gets in there, we need our body to know to fight him off. Well, he knows to fight him off because he doesn't have the right ID badge. Now, I have that. I can also add another structure to him, this purple chain right here. This purple chain, this is a carbohydrate chain. And what carbohydrate chains do, they aid in communication. Because if you think about it, so if I have my ID badge, but what if you see something inside your cell that isn't supposed to be there? Well, now you have to tell the other cells, you have to tell your immune system, be like, hey guys, there's something inside of here that I don't want. So they need to be able to talk to each other so they can get rid of foreign invaders and stuff. That's why we have carbohydrate chains. Now, if I add a carbohydrate chain to a peripheral protein, now this whole structure is called a glycoprotein. Glyco because of the carbohydrate chain, and it's attached to a peripheral protein. All right, I can also add a carbohydrate chain to a phospholipid. So now this entire structure right here, instead of it just being a phospholipid, this is a glycolipid because of the carbohydrate chain attached to a lipid. Now, regardless if you're a glycoprotein or a glycolipid, both of them still aid in communication because of the carbohydrate chain. That's what allows them to talk to each other, carbohydrate chains. Now I have a couple more structures. The next one, I'll draw it right here, and then I'll draw the lines down. He kind of looks like one of those swirly straws that you guys used to drink water out of. This guy is called an aquaporin. And you can imagine what aquaporins do, because aqua means water, por means hole. And so aquaporins, they're going to let water in and out. Water gets its own special structure because water makes up most of our bodies, and he also helps moderate temperature, and he also dissolves and transports substances. So he needs a fast pass into the cell, so that way I can get water in and out quickly. It's like having a fast pass to an amusement park. You get to skip the lines, you get to go right through. And then my last structure, we actually find him in the middle of my phospholipid tails, or my fatty acid tails. These guys, this is called cholesterol. And I know you guys have heard of cholesterol before. You're like, hey, cholesterol is not good. This is actually the good kind of cholesterol. What he does is he helps maintain fluidity. So your fatty acid tails, if I just kind of draw a simple picture, they're swaying back and forth, right? Well, at some point, sometimes, because they're moving around, they're in a living environment, these tails can start forming knots. And that's really bad because, again, if I'm trying to get this thing inside and there's a knot there, I'm not going to be able to get it in. This cholesterol helps keep those fatty acid tails. That cholesterol helps keep the fatty acid tails from knotting up, and they stay nice and separated from each other. And then my very, very last structure, we'll make him purple. We're going to put him over here on this side. This guy is called, I'm going to drag him all the way down here. This guy is called a protein pump. Okay, he is still an integral protein because he goes from one side to the other. But what he does is he is going to change shape to let things in and out of my cell. And if I want to change my shape, then that is going to cost energy, which is ATP. And so if I drew an example over here, so here's my protein pump. This is my door that's shut. And you guys all know that teachers have their ID badges. So if I come up here on a weekend, and here I am, and that door is shut, well, that door just doesn't automatically open. I have to have my badge. And so with this protein pump, this little guy comes up, but he's not able to go through until I have ATP. When ATP hooks to that protein pump, then that protein pump opens. So here's my ATP attached, which is going to allow my little green molecule to go through. Well, once that happens, once he goes through, I don't want to keep that door open forever. Like if I go up to the door and use my ID badge, it doesn't stay unlocked forever. So then that door is going to shut. So here's my green guy on the other side. This ATP, this detaches, and then it waits until I need it again. So that door just is closed. It opens when I apply energy, and then it shuts again. So this is different than the protein channel because the protein channel stays open for its entirety. Protein pump, I need energy to open it. So there's your phospholipid bilayer. You can see, we need things to get in and out. Those are my functions. There's my big structure of it. This is why it's arranged like that. And then here's all the other pieces. I hope that helps.